Good morning, good evening, good afternoon. This is Dr. Garais. This is BIO111. And this is a quick review of chapter 3, which is this a little bigger. All right, so what's chapter 3? We're going to be looking at the basic units of the body, and the term for regarding cells called differentiation means that a skin cell has DNA that tells it to be a skin cell, so it does skin things and makes skin proteins, and that differentiates itself from a cardiac cell or heart cell that will do different things. Now, because of the DNA or the basic blueprint of who you are, cells vary in shape, and size. And remember, size and shape is the morphology, it dictates the structure, which is physiology. So if you can see here, you could look at a nerve cell, also known as a neuron, and it's wire-like, and that's what the nerve cell does. You could see this, it propels electricity and signals in the same manner that a wire does. And you can also see the various sizes, and you can see the size of a typical red blood cell, and its shape is biconcave disc. And it's a lot smaller than the typical white blood cell here, which the biconcave disc has no nucleus. But if you look here, you can see this white blood cell is a little bit bigger than the white blood cell. You could also see some cells are flat, as in your epithelial cells, and it's like shingles on a roof or a shield. And then you look at here, contractile proteins, which are contraction means to get smaller. You could see these wires as well in striated muscular tissue, which is your voluntary muscle. Now, the major parts of the cell, and the best thing is to look at this picture. So what we did in class, we went over this picture and we started with the nucleus. The nucleus is the home or the brains of the operation, which is the control center of the cell. I want you to see the cell kind of like a mini human being. So this has a brain. The nucleolus has compact DNA in storage form. Yeah. And then the chromatin here is loosened DNA, which will be used by the RNA for copy and read in transcription and translation. The nuclear envelope is the covering of the nucleus, while this thing here, the phospholipid bilayer, is the cellular membrane, which covers the outside of the cell. Here, these little bumps here, those are called ribosomes. Those are the structures that make protein. Ribosomes that are associated with the rough endoplasmic reticulum, and this is the factory of making proteins. And they get the instructions from the DNA here, which the RNA transfers into outside of the nucleus. Here is the, well, this is still rough endoplasmic reticulum. You see there's still some bumps here, but if you look along here, these are all smooth. The rough endoplasmic reticulum makes proteins, and it makes proteins through the specific DNA code, and the DNA code codes to amino acids. Proteins are actually amino acids linked together by peptide bonds, and that's the job of a ribosome. And the rough ER is the factory for proteins. And the smooth endoplasmic reticulum is the factory for lipids and carbohydrates. You'll see here, this is the powerhouse of the cell, also known as the mitochondrion. The mitochondrion requires oxygen and glucose. The cell takes in oxygen and glucose. The mitochondrion then processes it through 3 processes that we're going to talk about later. But the end product is ATP, which is the metabolic energy that it requires to run the cell and to make the proteins that we need to make and the other products of the cell. And it keeps us alive. And when you think about it, I require oxygen, I require glucose to live. And of course, the byproducts will be carbon dioxide, H+ ion, and ATP, which gets used up. And we talked about before how there are certain bonds that are high powered, and the phosphate bonds in ATP are high powered bonds. Now, there are also lysosomes and peroxisomes, some meaning body, peroxide and lyse, they break down things, and they need to be packaged by here, which is the post office of the cell, this purple item right here, and that's your Golgi apparatus. The function of the Golgi apparatus is to package things so that it will make sure that those harmful products like peroxides and lysosomal enzymes that break down things stay in their package until they're needed. The package can be opened and utilized inside within the cell, or it can be released as secretory vesicles to protect the cell against foreign body and or pathogenic material. The yellow part here, the jelly-like structure that gives the cell its shape, along with some microtubules with these microprotein strings here, that gives the shape and form. And forms like little pathways, kind of like little highways, so that the packaged vesicles, such as the peroxisomes and lysosomes, can move around. Another function of this as well, as you learned in laboratory, is mitosis. When the DNA duplicates and then the cell starts to separate, these microtubules elongate and are controlled by these two things right here, which are compact microtubules, and these things here are called centrioles. Upon prophase, which is the first part of mitosis, the centrioles move to different poles and then start connecting all the microtubules. And then in metaphase, all the genetic material line up midline. Anaphase, they start to pull apart. And of course, telophase, that's when we have cleavage and separation of this single cell that got doubled into another cell. Lastly, the microtubules can also be used for motion and or protection. You have here microvilli and here cilia, which move things. And we talked about the example of movement of mucus through your respiratory tract, so it can go up and out and into the garbage. And the microtubules, we talked about this flagellum here, this tail that can help move the cell. And the classic example there is sperm spermatozoa, also known as sperm cells. So we covered all that, and we can go through all of these, but let's now talk about the cell membrane. It's the outer boundary of the cell. You also have a nuclear membrane that covers the nucleus, but the outer membrane is called your cell membrane, and it's made up of a phospholipid bilayer. The inside of this layer is made out of fatty acids, which is the lipid part, and the phosphorus heads here are hydrophilic. That means here loves water, but in the middle it's hydrophobic or doesn't like water. So all the water and water pressure stays constant on the outside. All the water pressure intracellularly stays constant and stays on the inside. But we'll show you how this is a semi-permeable membrane, how some things get in, some things get out, and it is by pure choice of how the cell does that. So it's semi-permeable, selectively permeable. Phospholipid bilayer, cholesterol, yada. Let's talk about it here in a better view. So this is your bilipid layer, or bilayer, and this is the outside of the cell, which is extracellular fluid, and this is the inside of the cell, which is intracellular fluid. These things are proteins. You have integral proteins here. You also have proteins here. Now, these proteins help stiffen up this mosaic model of your bilipid layer. The proteins, carbohydrate chains, and even sugar. So there's carbohydrate chains, sugar chains, and protein chains on the outside, and they're utilized as a chemical marker to distinguish self from non-self. So it's kind of a way where your immune system knows who the good guys and bad guys are. We also talked about how hormones are like a key to open up these doors. Remember, semi-permeability. So let's say this thing right here is a receptor. So it's a lock. You have something like a key, which is like insulin. It opens up the lock, which is the insulin receptor. And then this door can go open nice and wide so that C6H12O6 can then go inside the cell. Now we know about diabetes. either my patient doesn't have insulin, which is the key, or they don't have a very good lock, which is your receptor, then glucose won't be able to go inside the cell. If glucose doesn't go inside the cell, it parks itself on the extracellular fluid, which isn't a good idea because then the glucose starts doing damage. The analogy in the classroom is if you go to the gas pump, it's not putting the gasoline or the glucose inside the cell where it belongs, where it can be processed by the mitochondria. Instead, it floats around. That's like putting gasoline on your car seats and on the hood of your car, which of course, obviously is not a good idea. Talked about ribosomes, talked about all that. So Golgi, the rest of these slides go over the exact same thing that I was talking about and in a little bit greater detail, but it's all the same thing. See here, this is your mitochondria. I don't know why they labeled, here's of course your lysosomes. The lysosomes are here. They have things inside there that will lyse or break down. So it's a body, somatic, that will break down something. So a peroxisome is a some or body or cell or part of the cell, right? And the parts of these cells I failed to mention, they're called organelles. So you have to start to look at the cell as being kind of like a mini human being. Microfilaments, microtubules, and you can see here how the microtubules form a scaffolding so that everything can be in its own place. And it then forms a nice little scaffolding so it's like a highway so that things can go in and out. And again, the cytoplasm and the microtubules, beautiful both A and B question, give shape to the cell. And we already now know form or shape or morphology leads to function. Cilia, flagella, so it's all the same thing. So now the next part of this lecture is how do things move around? Things move around either through energy or they can move around passively. If something moves around passively, that means what? Because it does not require ATP. Here are the 4 examples of things that move passively: diffusion, osmosis, facilitated diffusion, and filtration. All 4 of these move particles or move fluid from an area of high concentration to an area of low concentration. Diffusion is the classic example. Osmosis is just like diffusion, but it's going through a semi-permeable membrane. And an example of the semi-permeable membrane is our nuclear envelope, and of course, the bilipid layer that we just discussed. Facilitated diffusion still moves from an area of high concentration to low concentration, but it goes by a little bit faster than your typical osmosis or diffusion. And last but not least, filtration. That's when you want to filter something. So the analogy in class was a coffee maker where you put all the powder on the top and then what gets through? The coffee. Okay, and all of these things, diffusion, osmosis, facilitated diffusion, and filtration are all passive mechanisms, meaning to say it does not require ATP. But what does require ATP? Let's go fast forward a little bit. It's called active transport. Active transport is coming from an area or a gradient of lower concentration to an area of higher concentration, which is of course means that it is against the flow or against the gradient, or it's also going uphill. So these things, there are certain carrier molecules that are inside our bilipid layer that move things against the gradient. And we talked about sodium and potassium and chloride and ions. This is an active transport, so it requires ATP. Another thing that requires ATP is doing stuff like this. Remember the Golgi apparatus that has to package things and make their own little package and then secrete them out? Well, there's 3 of those types of functions called endocytosis, penocytosis, phagocytosis, and receptor-mediated endocytosis. Don't need to really try to memorize all this. Just know that what? If I package something in a cell from the inside, which is endocytosis, that means what am I doing? I'm moving things into a vesicle or a package, just like a peroxisome or a lysosome, and that requires energy. So those things require from from a gradient from low concentration to high concentration. This next part here talks about tonicity, goes hypertonic, isotonic, and hypotonic solutions. Compare this to your the exercise on your Unit 1 laboratory that we just performed today, when we're talking about how it affects the red blood cells. And here are some lovely examples which you guys chose from, and I hope you cited your textbook. Okay, so go over that as well. That's a lovely test question. Okay. And I'm not really going too much on cell cycle. Nice to know, but definitely for laboratory, you need to know the phases of mitosis. Mitosis is cell division, and what's it going to do? If it's a somatic division, meaning if I start off with a skin cell, I should end up with a skin cell. And these are the 4 phases of mitosis, which you should already be familiar with: prophase, metaphase, anaphase, and telophase. The notes go through all of that. And last but not least, how do we differentiate? How does the cell know what to be? And of course, we know about the DNA, the blueprint of who you are. And then we take these things called stem cells. Stem cells, progenitor cells, totipotent cells, and pluripotent cells. These are the starters, right? And then they can be certain things. So you can have a progenitor cell that's going to start out that's going to be something. Then it'll be what? Committed. Once they're committed means what? I'm definitely going to do this. I can have a totipotent cell that can be what? Any type of cell. And a pluripotent cell has a what? A limited amount of leeway on what's it going to be. So toti, think what? Anything. Pluri, plural, something. So that's my quick review for chapter 3, and I'll also put later on today some sample questions. Thanks for your plan, and I'll see you in. Have a wonderful and highly uneventful day. Yes.
Can I Ashley for a moment?
Miss Ashley, you here? Or she left already? She left already.
All right, thanks.
Stop recording.